Hello everyone and welcome back to our channel Biodiversity and Conservation. I am Subhalakshmi Raut from Ambika Prasad Research Foundation, India. We are continuing our UGC NET examination series for environmental science subject and this is the 27th part of Unit 3 Environmental Biology. Today we are going to discuss about one of the biogeochemical cycles that is nitrogen cycle. Nitrogen cycle is an example of gaseous biogeochemical cycle, and the main reservoir of nitrogen is atmosphere, and it is the most abundant element of the atmosphere. And nitrogen is an essential element for living organisms because nitrogen is a component of almost every living matter, that is amino acids. They are formed from nitrogen, which forms the building blocks of protein. And nitrogen is also present in the nucleic acids, and which gives us DNA and RNA. So it is an essential element for our protoplasm, and it is required nutrient for all living organisms to produce the complex organic molecules. And there are some steps in nitrogen cycle that helps in the cycling of nitrogen. They are nitrogen fixation, ammonification, nitrification, denitrification, so and assimilation also. So these are some of the steps which we are going to discuss. Here in this diagram you can see the nitrogen in the atmosphere is fixed in the process of nitrogen fixation by the help of some nitrogen fixing bacteria, which converts the N2 into ammonia, NH3. Then the ammonia is nitrified through nitrification with the help of nitrifying bacteria into NO2 minus. They are the nitrates and nitrites. And ammonification is the process where decomposers break down the dead decaying matter into ammonium ion. That process is called ammonification. And then next step is assimilation, where the nitrates produced from nitrification are absorbed by the living matter, may it be plants, animals, or microbes. Then there is the last process, that is denitrification. It is a process in which the nitrogen, molecular nitrogen, is um, given back to the atmosphere by breaking down the nitrates. This also have some denitrifying bacteria. They play an important role in this step. So basically this is the brief. Now we are going to discuss the steps one by one. First step is nitrogen fixation. Here nitrogen fixation can be done through physical or non-biological process or through biological process. As we know, nitrogen cannot be utilized by plants in its native elemental form, so it must first be fixed or combined with other elements like hydrogen or carbon or oxygen. Then only it can be used by the plants to produce different products which will be helpful for other consumers. So it is a process of nitrogen fixation where um, the nitrogen available in the atmosphere is fixed into nitrites or nitrates. So in case of non-biological or physical nitrogen fixation, we have industrial and natural process. And in case of biological nitrogen fixation, we have non-symbiotic or symbiotic process. So we are going to discuss one by one. First one is biological fixation of nitrogen. Here actually it occurs in both autotrophic and heterotrophic organisms, and it can also occur in aerobic or anaerobic zones of the ecosystem, and it can be done by symbiotic or non-symbiotic means. So in case of symbiotic nitrogen fixation, it occurs in plants that have nitrogen fixing bacteria, and they are present in their tissues, um, and these bacteria can survive independently, but like legumes, but their life together is clearly beneficial to both. So they remain in symbiotic association with each other. So first case in symbiotic nitrogen fixation is legume symbiosis, where the root nodules of legumes like peas, beans, etc. Um, they are they fix nitrogen for the plant um, by being present in the root nodules of the legumes, and in these leguminous associations, the bacteria usually are rhizobium species, but the root nodules of soybean, chickpeas, and some other legumes, um, they are formed by some small celled rhizobia known as bradyrhizobium. So in this process, actually, the root nodules have infection thread, um, which is constructed by the root cells, um, and it is only formed in response to the infection. So when the infection thread reaches a cell deep in the cortex, it bursts and the bacteria are engulfed by endocytosis into the endosome. At this stage, cells go through several rounds of mitosis without cytogenesis, so the cell becomes polyploid, means it has more nucleus than but have only a single cell. So uh, with this continuous process, the cell becomes larger in size, and after some time it bursts. Um, so the rhizobia are engulfed into endosomes within the cytoplasm, and then the cortex cells begin to divide rapidly, forming a nodule. This is the node factor. So um, this nodule actually helps in nitrogen fixation. So how it's formed is discussed here, and the bacteria live symbiotically in the nodules, obtain food from the root tissue, while the plant receives nitrogen fixed by them. And there are some other associations also, like cyanobacterial associations. Here the association is with azolla, where the cyanobacteria are present in the leaves, and um, are important for nitrogen inputs in rice paddies, etc. And the next is the non-symbiotic fixation of nitrogen. Here the fixation of nitrogen is done by free living nitrogen fixers. Some include fixation by soil and water bacteria like azotobacterium, aerobic bacteria, and clostridium anaerobic bacteria. They are widely distributed in the soil, and they help in fixing the nitrogen of soil, and build up amino acids from which proteins are made. When these bacteria die, their proteins are released into the soil, so we get the proteins from the soil. There are also fixation by blue-green algae and yeast. So all these are some examples of non-symbiotic fixation of nitrogen. And in non-biological nitrogen fixation, industrial nitrogen fixation, um, lightning, combustion of fossil fuels, all this comes under this. Industrial nitrogen fixation is the process where under great pressure at a temperature of 600 degrees Celsius, with the use of iron catalyst, hydrogen and atmospheric nitrogen can be combined to form ammonia. So this process is called Haber-Bosch process, which is used to make fertilizer and explosives. Next one is combustion of fossil fuels. Through the use of automobile engines and thermal power plants, we release of various nitrogen oxides and into the air, which gives us available nitrogen for our consumption. And the formation of NO from N2 and O2 due to photons, especially through lightning, also can fix nitrogen. So, and we will go to the next process. The next process is nitrogen assimilation. In this process, the inorganic nitrogen, that is nitrates or nitrites and ammonia, is absorbed by the plants through their roots and then is converted into nitrogenous organic compounds. So these nitrates are first converted into ammonia, which combines with other organic acids to form amino acids. And the amino acids which are formed are used in the synthesis of proteins, enzymes, nucleic acids, etc. And after the plant produces these compounds, when the plants are consumed by other animals or other consumers, a part of the plant protein passes into them by the process of consumption in the food chain, where they get converted into animal protein, while the rest gets decomposed after death and decay of the plant body also. So the protein is eventually broken down into nitrogenous waste, and these wastes go back to the soil and to the atmosphere eventually. So in this process, the nitrogenous compounds are absorbed or used up by the plants or animals. This is called nitrogen assimilation. Next process is ammonification. Here, when a plant or animal dies, or an animal expels waste, the initial form or the nitrogen in the tissues is in the form of organic nitrogen. The release of ammonia from organic nitrogenous compounds is termed as ammonification. Proteins and other nitrogenous compounds of living and dead organisms which find their way into the soil are decomposed by some microorganisms, and these compounds, the organic nitrogenous compounds, are hydrolyzed by some proteolytic enzymes into amino acids and similar compounds, and the amino groups are split off to form ammonia. And these proteolytic enzymes are found in some fungi, some actinomycetes, and in Pseudomonas, Bacillus, etc. So the ultimate product of proteolysis are amino acids, and the amino acids are deaminated under aerobic conditions with liberation of ammonia. So if protein decomposition occurs under anaerobic condition, the amino acids are converted into amines and related compounds. So these reactions are most commonly brought about by Clostridium species. Eventually, the amines are oxidized in the presence of air with the liberation of ammonia. Similarly, in case of man and animals, the urea present in the urine is decomposed with the liberation of ammonia by several microorganisms, especially by Micrococcus species. Ammonium ion is toxic even at low ion concentration and is therefore never allowed to accumulate. So most of it is assimilated by soil organisms or microorganisms, and some of it is used by plants as a source of nitrogen, and microbes utilize the organic nitrogen rich substrate into their metabolism and in the process release ammonia. So these microbes actually help in excreting out the extra nitrogen, and this process eventually is called ammonification because of the final product of ammonium ion. Next process is nitrification. Uh, Classically, this process refers to as the oxidation of ammonia into nitrate via nitrite. So the bacteria that carry out this process are called nitrifying bacteria, and they are aerobic and chemoautotrophic. That means they produce food in the presence of chemicals, and belong to the genera of nitrosoph or nitroprofix, and gain the energy from the inorganic nitrogen reactions and gain their carbon from carbon dioxide. So in the first step, the oxidation of ammonia to nitrite is called nitrosification, where NH3 is converted into NO2 minus ions plus water and H plus ions when NH3, that means ammonia, is mixed with oxygen. And this process is accompanied by the bacteria of genera Nitrosomonas, Nitrosococcus, Nitrosospira, and Nitrocystis. And the nitrites so formed by these organisms are toxic to plants as well as the organisms forming it. So the nitrites are removed by further oxidation to nitrates by the bacteria of genus Nitrobacter. Here, the nitrite combines with oxygen to form nitrate, and this um, process of oxidation of ammonia is called nitrification because of the production of nitrites and nitrates, and these are done by some bacteria species here through the help of some enzymes like ammonium monooxygenase, in where the ammonium ion is or ammonia is converted into nitrite, and in nitrite oxidoreductase, where the nitrite is converted to nitrate. Now the last process is denitrification. where the nitrogen can, however, be permanently lost from the cycle by biological process of denitrification. It is the reduction of nitrates back into the largely inert nitrogen gas, that is N2, thus completing the nitrogen cycle. So here the nitrates are reduced via nitrous oxide and to nitrogen gas, and in this the nitrate is replacing oxygen as the electron acceptor in the biochemistry of the cell. So here the nitrate is reduced to NO2 um, with the help of the enzyme nitrate reductase. Then NO2 is reduced to um, hyponitrite uh, with the help of nitrate reductase, and then we get three products by the reduction um, of hyponitrite through the enzyme hyponitrite reductase into NH2, N2, and N2O. And N2O eventually then breaks down into molecular nitrogen N2. So in this process, many different kinds of facultative anaerobic bacteria uh, are involved, like Pseudomonas, um, Paracoccus, Bacillus species, Micrococcus denitrificans. There are several bacteria that helps in this process, and um, the Micrococcus denitrificans is a facultative autotroph that develops aerobically or anaerobically with either organic compounds or H2 as the energy source and oxygen or nitrate as the electron acceptor. There is also Theobacillus denitrificans. Um, it is also a chemotrope that helps in denitrification. So there are some importance of nitrogen cycle. Why actually nitrogen cycle is important? So the cyclic process of nitrogen cycle balances out the availability of chemicals and all the different products, organic compounds um, available for use, and there is also a critical role of several microorganisms in the nitrogen cycle, like the cyclic flow of nitrogen through ecosystem requires a balance of bacterial action, and these microorganisms helps in regulating the nitrate concentration in different pools, and so understanding the role of these microorganisms in these diverse ecosystems is essential, and not only for understanding the nitrogen cycle but for the management of the emerging environmental problems. We have to understand the role of microorganisms in the ecosystem and nitrogen cycle, and even if we lose some organisms due to extinction or other factors involved in this cycle, it will eventually harm the cycle, and that there is also some loss to nitrogen cycle also. Now we will discuss briefly about the differences between nitrification and denitrification. The nitrification process is obligately aerobic. It is chemoautotrophic and requires carbon dioxide for carbon source. and gain energy from inorganic reactions. And the nitrifying microorganisms belong to well-defined taxonomic group. It is a reversible process and inhibited by excess carbon. But in case of denitrification, the process is facultatively anaerobic. It is heterotrophic in nature and requires organic carbon for carbon source, use organic carbon for energy source, and it is a variable group. Many members from different genera are present here. It is a non-reversible process and requires organic carbon source. Thank you. And this is all from me today.